All right, so in your current book, they cover adaptive as well as disorders all in one chapter. But I split them up into 2 topics, right? But this next chapter is a whopper, right? So much so that we spend our last bit of the semester on it, right? This is it. This is adaptive immunity. And then, of course, disorders. And so adaptive, as we've said before, right, refers to this change over time, right? So this is the really specialized component of our immune system where we're really going to adapt and change over time and get really specific in our interaction. And one of the main parts of our adaptive immune response is an antibody response. So we're going to look at these foreign substances in a little bit more detail, what we refer to as antigens. And as it applies to especially antibodies, antibodies only bind to a small portion of an antigen most of the time. We refer to these as epitopes or antigenic determinants. So we're going to look at the nature of antigens first, and then we're going to go into beginning talking about antibodies and their diversity and their abilities. So as the name implies, right, antigen means antibody generators. And again, the most common response of our adaptive immune. But that's not the only response. We do have a cellular response that we're going to talk about. So some people have proposed, instead of calling antigens, which can be confusing between antigens and antibodies sometimes, that we should call them immunogens, right? Because to refer to the fact that you have an immunological response, right, that's generated from these foreign substances. I have not converted yet. I still say antigen. I don't know. When you learn a particular way, right, you tend to keep doing it that particular way. And so for me, we called them antigens, right? And you're still going to see that term, right? Not everybody has converted. But it's an important thing to note that, you know, this term came from the production of antibodies, right? This ability that we saw that, oh, we make antibodies against foreign substances. But that's not the only thing we do, right? Antibodies is not the only response, right? We do have, in certain instances, a cellular response. That's really important. So antigens, some of them, proteins and different polysaccharides, can induce a string response that we'll talk about later. And that's because they're highly repetitive molecules, which means that lots of antibodies can bind to the same molecule at the same time. Right? That's what they mean by string response, right? So they're all kind of lined up in a string. Lipids and nucleic acids often do not, and they're not as immunogenic as proteins and sugars are, because there's not a lot of variability in those molecules. So as we'll see in this animation, as I said, the antibody doesn't bind to the whole antigen, but only a small portion of it that we commonly refer to as an antigenic determinant, or I prefer the term epitope.
Antigens are macromolecules, usually of molecular weight greater than 10,000, such as proteins and polysaccharides. They are recognized by the immune system as foreign. Individual antibodies are not made against the entire antigen molecule, but rather to particular chemical groups on the molecules known as antigenic determinants or epitopes. Many different antibodies can be made against a single antigen, each antibody reacting with a different epitope. Complex structures, such as the surfaces of bacterial cells, may have many different epitopes. Each different antibody binds only to the correct epitope.
So this is the important thing about antibodies, right? Is that they are very specific as to what they bind to. In the case of a very complex antigen, like this bacteria or even some proteins, right? Proteins are made up of so many different amino acids that we can make antibodies that bind only to small portions of these very large antigens, right? So this antibody is only binding to this specific antigenic epitope on the bacteria. This other antibody is binding to this, and so here in this diagram, we have 4 different antibodies we've made against the same bacteria, right? So we're multifaceted in our approach. It's also referred to sometimes as polyclonal, poly meaning many. When we use antibodies for testing, though, sometimes outside the body, right, when we use antibodies to help us identify things, we utilize a purified set of antibodies, commonly referred to as monoclonal, which means it's a single antibody binding to a single site on the antigen. And so, again, these are very beneficial because we know exactly what it's binding to. And there's a lot of rapid tests out there that use this type of technology of antibodies outside the body to help detect because of this level of specificity. So how many of you guys have taken anatomy and physiology too? Right? You've done blood typing, right? What did you add to the red blood cells to figure out what antigens they had? You added antibodies. You added known antibodies. You added the anti-A antibody to a well of red blood cells to see if those red blood cells had the A antigen. If they did, the antibodies bound to those red blood cells and clumped them up, called an agglutination reaction. But we were able to know because the antibodies only bind to that antigen, right? To that molecule on the surface of the red blood cells if it's present. You didn't see clumping, right, if you didn't have that antigen. Like my blood type is boring. I'm O. So when you add the anti-A, nothing happens. When you add the anti-B, nothing happens, right? Because I don't have either one of those antigens. So you put those antibodies there, there's nothing for them to bind to. There's no reaction. So these types of reactions happen inside of our body too, right? If we have an antigen and we have antibodies against it, it's going to specifically bind, right? But at this molecular level, right, we're talking about very specific molecular recognition of pathogens. So for instance, E. coli, a particular strain of E. coli called 0157, that particular strain, unfortunately, has gained the ability to produce the sugar toxin, right? I've talked about it before. We don't know exactly how, but it got it from Shigella, right? It got that DNA on how to make that toxin. Well, that particular strain of bacteria also makes a particular group of sugars in its lipopolysaccharide. We have antibodies, and that's what the 0157 refers to. That antigen, that specific group of sugars on the surface of that bacteria, we can use antibodies to detect it and identify it. It'll only bind, those antibodies will only bind to that set of sugars and let us know, oh, that's E. coli 0157. That's bad news because he produces the sugar toxin. Make sense? So these are very powerful substances because they are so specific in their recognition of antigens. Their structure is very important to their functionality too. So notice they're drawn as little Y's here. We're going to dive deeper now into that molecular structure, right, and the different components and how they work. So you need to, if given a diagram, be able to label these different regions, right? You need to identify the functions of the constant and antigen binding regions. There are 5 different classes of antibodies. You need to know the differences between the classes and what they do. And then there are 6 different protective outcomes that can happen from antibodies binding to antigens. So a lot of stuff related to antibodies. So let's first start out with the structure itself. I've never been 100% happy with the diagrams in the book. I've gotten them over the years to change it a little bit, but I'm still not. They did separate them out, right? But I find that even sometimes they're kind of confusing. So I'm going to work with my own diagram. So I could put this diagram on the test, right? And notice I have all the different parts labeled with letters, right? So I could basically ask you, you know, with what letters are what, right? So antibodies in general, right, all are made up of 4 protein segments. 2 of them, 2 identical pairs is what happens. 2 of them are really short, and 2 of them are really long. The 2 really short ones, of course, because they're shorter, tend to be lighter, and so therefore they're called the light chains. So you have 2 of those denoted in blue here, and then the longer chains in green, what we call the heavy chains. Those 4 pieces, right? The 2 heavy chains are identical, the 2 light chains are identical. Those 4 pieces bind together in this particular arrangement to give this important structural arrangement to this protein, right? This characteristic Y structure. Now, of course, the way we have it drawn, y'all, is like drawing people in stick figures, right? It's a very simplistic representation of a person, a stick figure, yes? All right, we're playing like Hangman or something like that. These are very large, somewhat globular molecules, but overall, right, you can search on the internet and see 3D representations of antibodies. But again, overall, they look like a Y, right? They have what we sometimes refer to as 2 arms and a stem, right? The arms are important in that they bind to antigens. So these A's and B's are representing the antigens, and more specifically the antigenic epitopes, right? That small piece of the antigen that the antibody is binding to. C down here, where C is, this is where our immune system interacts with the antigen. Excuse me, antibody. I probably should start saying immunogen and antibody, right? It would be easier. You get tongue twisted. In order to achieve this, right, in order for antigens to bind up here at the antigen binding sites, and for the immune system to be able to interact with this stem portion, you'll notice that they're different, right? First of all, the stem is only made out of heavy chain, right? This portion up here that interacts with the antigen is made from light chain and heavy chain, the combination of these 2. And again, this molecule is a mirror image of itself, right? If you split it down the middle, the 2 light chains are identical. The 2 heavy chains are identical. Still with me? Okay. So the next thing that we look at, too, is that you can actually break an antibody into these 3 pieces if you bust them at the hinge range right here. You're going to end up with an A and B fragment, and this is what they did to antibodies when they were first studying them. Right? They broke them up into their respective pieces. When they broke it here at the hinge region, they ended up with an A and a B, which again, they're identical to each other but mirror images. And they found that this fraction, this A and B fractions, bound to the antigens. So this is referred to as the FAB regions, right? Which works out too, right? Because I have them labeled A and B here. But the AB actually stands for antigen binding. The C portion, the C actually stands for the fact that that fraction crystallized. But we just commonly refer to this fraction as the FC fraction or FC region. And this is the portion that the immune system interacts with, right? So immune cells or immune components interact with this portion of the antibody, the FC region. So again, to help us in identification, I labeled this C. It works out. So the next thing is shown in those other diagrams. You'll notice that they color the light chains and the heavy chains a little bit differently up here in the antigen binding region. That's because for each antibody that your body produces, if it's made by a different B cell, this region of both the light chain and heavy chain is different. This is what gives antibodies their uniqueness. This is the variable region. And both the light chains and heavy chains have this variable region. And this was what makes each antibody unique. So in that previous diagram where we had, 4 different colored antibodies, that was to denote that each one of those antibodies, its variable region is different, and that's why it binded to a different antigenic epitope. Now, if you look at the structure of the antibody, though, light chains bind to heavy chains, right? Where do they bind? Here, here, and then the 2 heavy chains bind with each other right here in the hinge region. In order to always have heavy chains and light chains binding with each other and heavy chains with each other, the protein, the molecular makeup of the protein in these regions needs to be consistent. Right? Because if you don't make that protein the same way in these regions, the antibody is not going to form the right way, right? Light chains aren't going to attach to heavy chains aren't going to attach to heavy chains. And you're not going to get this hands, right? These arms and stem structure. The functionality of the antibody wouldn't work. So that's why this blue region is referred to as the constant region. So both heavy chains and light chains have constant regions, right? The light chains just have one segment shown here, where the heavy chains, most of them are constant. So if we were to use this labeling system, right, the FAB regions are this portion and this portion, which are labeled A through H, right? The FC region is this portion here, which is labeled I through L. The constant region, remember both the light chain, right here, this portion, and starting here for the heavy chains, on down is all constant region. So E through L. The variable regions is just the first portion of the light chain and the heavy chain on both sides. So our variable regions are A, B, C through D. Antigen binding sites are denoted here by M and N. And why do I have O down here? Antibody? Nope. What is that region? Why is that region important? Why is the O region important? What happens there? Yeah, that's where the immune system interacts with the antibody, right? Because this portion up here, basically the arms, are interacting with the antigen. So as you see here, right, here's a bacteria. Notice the variable regions of the heavy and light chains denoted in blue here are binding to the bacteria. Therefore, the stem, right, the FC portion is sticking out. You see that? That's the portion that our immune system is going to interact with. Right? So phagocytic cells can grab onto it, right? Antibodies can be opsonizers just like C3b. Our phagocytic cells, for some of our antibodies, they have receptors for the FC region. They're going to grab onto it and gobble up that bacteria. That's one of the protective outcomes of antibodies is opsonization, tagging for destruction by phagocytic cells. And it's all due from the fact that the antibody specifically binds to that antigen at a molecular level, right? Right down to that specific group of polysaccharides that E. coli 0157 makes, right? Or to the specific toxin produced by tetanus, right? We make several antibodies that can bind to that toxin. So here's an example, too, of a particular class of antibody, right? So antibodies are also referred to as immunoglobulins, right? A product of our immune system, globulin referring to that protein globular structure that it has. And then each of the classes of antibodies is given a letter designation. This one, right, IgE, that class of antibody, binds to receptors specifically for the FC portion of that class of antibody, of IgEs. This receptor, unfortunately for some of us, is found on mast cells and basophils. So when antigens then bind to the antibody, it stimulates the mast cell or the basophil to degranulate. Anyone know what substance that's very common in mast cells and basophils that can cause an inflammatory response? Histamine. This is why I always tell my students to remember E for Erica, because Erica hates her IgEs, because these are the ones that are responsible for the allergic response, which unfortunately I'm allergic to most of the pollens outside. So right now, right, sinuses are screaming at me, even though I've taken all my allergy medicine. It's just more than my system can handle. So the next thing that we're going to look at is protective outcomes, right? How do antibodies protect us? Well, they bind to specific antigens, but how is that protective, right? So what? It binds, right? In the case of IgE, we're usually not so happy that it binds, right? Because of the response that we give. So in this case, you've got a virus coated in antibodies and a toxin coated in antibodies. This stops the virus from attaching to our cells when it's coated in antibody. So why the flu shot could help you, right? Because you could produce antibodies that bind to the virus first before it attaches to your cells, which is the first thing it's got to do to gain entry, right, is attach. Well, can it attach if it's coated in antibodies? No, we've created a barrier between the virus and ourself by coating it in antibody. Same thing with toxins. Toxins usually have to interact directly with our cells. If it's coated in antibody, it can't interact. So this is said to neutralize the virus or the toxin. So this is a term specifically for antibodies binding to viruses and toxins and stopping them from being able to act. This is referred to as neutralization. So this is one of the potential protective outcomes of an antibody binding to an antigen. So if it's binding to a virus or a toxin and it stops it from attaching to our cells, then it's said to have neutralized them. The next one, so you see a bacteria coated in antibodies, and then some cell is grabbing onto those antibodies. What type of cell do you think that is? It's a phagocyte. Any guesses as to which one? Neutrophils are the most common, right? And so that would be a very good guess. Except there's a clue here. Look at the nucleus. Does that look like a neutrophil nucleus? No, neutrophil nuclei, remember, are multi-lobed. That's not multi-lobed. So this is probably a macrophage, or maybe a dendritic cell even. So what's happening here is that phagocytosis is happening because the antibodies are attached to it. So these antibodies are tagging it for destruction. What do we call this when it gets tagged for destruction by a phagocytic cell? Opsonization, right? The same thing that what complement protein does? C3b, which is that question. So not all our antibodies can aid in this, right? Only one class that we're going to talk about do our phagocytic cells have receptors for, have FC receptors for, because remember it's grabbing onto the FC portion of this antibody. You see that? Because the antigen binding is happening with the variable regions here. So here is the FC portion. It's grabbing on. So that class of antibody is IgG. That class, this FC region, is recognized by our phagocytic cells. So, which protective outcome are we showing here? We've got antibodies binding and interacting with a protein, and the outcomes we're going to get from that interaction is more opsonization by maybe C3b. We'll get an inflammatory response, and we may even get lysis of foreign cells via the MAC complex. So what are we talking about? What system here? What has C3b in the MAC complex? What do we call this? Complement, right? This is complement activation via the classical pathway, right? Some antibodies, when they bind, they can interact with complement proteins and activate complement. Next one is you've got some bacteria here with flagella. And then antibodies bind to the flagella. The flagella on a bacteria spin like a propeller on a boat, right? So this really happened to me too, right? Grad school, going down the bayou, helping one of my colleagues collect samples for studying parasites, actually Miss Brock to be specific, right? And we stop. The motor stops. We're like, what the heck? We pull the motor up. There's a sweatshirt wrapped around the propeller. Right? That's why we stopped going. So we had to take the sweatshirt off the propeller, right? So that we could keep going down the bayou and pick up our fish samples for her. What happens to the bacteria, right? They've got a propeller. It's called the flagella, right? And antibodies stick to it. That's basically like a sweatshirt wrapping up the propeller on that boat, right? So imagine, right, their propeller gets all tangled up in antibodies. What's going to happen? They're going to stop moving. Is that beneficial to us? Yeah, because remember, what do we have that helps protect the openings of our body? Mucus. If they can swim through the mucus, we can't stop them, right? But if we've got antibodies, right, that bind to them and stop them from moving, then we can keep them trapped in the mucus and we can make them move where we want them to go, which is out. So this is very simply called immobilization, right? We're immobilizing them. Microorganisms also tend to have protein appendages on their surface that help them, that attach to us. If there are papillae that are specifically for that, they're referred to as fimbriae. And so in addition, if antibodies bind and stop them, therefore, from binding to us, that is beneficial as well. And again, this can be very beneficial in our mucus, right? To have antibodies there that bind to them before they bind to our cells. So again, keep them trapped in the mucus, moving them out the body. Is this my last one? Yeah. Humor me for 1 second for this last one. So this is the one that you see in anatomy and physiology lab. When you take antibodies and add it to the red blood cells, you cause this cross-linking, because antibodies, at the very least, can grab on to 2 bacteria at the same time, and then multiple ones, so you get this cross-linking effect. When it's very large antigens like your red blood cells or a bacteria, that reaction, when it happens outside the body, is referred to as an agglutination reaction. So that's why they call it agglutination, right, in A&P lab. There's a reason for this. If you're dealing with really small soluble substances, because they're small and soluble, which means they're in solution, you would normally see them. So there are some tests that we perform with, say, proteins in solution or in a gel that you can't see them. But when you add the antibodies and the antigen and they interact and cross-link like this, it'll cause what's called a precipitation reaction, where you can literally see, because it's such a large molecular complex, you can see it precipitate out of solution or form a line in a gel. It's really cool. One of our antibodies that we're going to learn about next time is really good at this, and this is the one that you use for the ABO blood typing. It's IgM. It has 5 Y-shaped components to it. And so it's really good at cross-linking and agglutination. So next time we're going to go over these 5 different classes, and we're going to review some of this stuff. So next time we're going to do a little bit of clickers. Or, you know, your phones. We don't do the clickers in this class. Oh, there's one more outcome. That's okay.